Hi, and welcome to Live for the Cure. My name is Aaron Foreman, and I'm the Vice President on the board at the Live for the Cure. And in this video, I'll discuss the three overarching problems in delivering gene therapy to children with large chromosomal deletions. In an attempt to discuss the topics in a more understandable way, I will use similarities to something we use every day: computers. Computers are part of almost every aspect of our lives. Computers are found in our cars, our cell phones, and even our fridges. Comparing the human body to computers is actually not such a far stretch. In fact, it's interesting how computers and software are made very much in our likeness. They take over all the tasks that we find mundane and difficult. They calculate, store, remember, and they're present in very special use cases such as arts and sciences. Fundamentally, they are the expression of us. The software that we use on these devices are developed in a very controlled environment. There are rules on how they present on screen and render data for our review. There are rules on how many things it can do at one time or how many resources are required to complete a task. As a programmer, I am confined to the language that I'm developing my software in. That programming language gives the guide and the tools to create applications. When we look at the human body, we're naturally presented with a radically different set of rules, but those rules are also a guide. The fact is, we're the most complex biological computer systems on the planet. Just like computers, we have a hard drive, our brains, and when we work on something in the here and now, we're using what is similar to RAM within the computer, or random access memory. Human being software, or chromosomes, are thread-like structures inside the nucleus of our cells that carry our genes or specific genetic makeup. While 99.9% of our genes are exactly the same across every human being, it is the 0.1% that gives us our variations, our uniqueness. Inside the nucleus, the DNA looks like a ball of yarn, which are all the chromosomes intertwined. As we dive deeper and look at a specific chromosome, we see that there is a pair. Each member of the pair has a short arm, or a P arm, and a long arm, or a Q arm. Diving deeper and looking at a piece of one of those arms, you can see the famous double helix structure that composed of double-stranded molecules of nucleotide acids. The structure's building blocks are composed of base pairs. These base pair sequences make up genes that generate proteins or enzymes, depending on their purpose, which are the building blocks and functioning of all the systems in the human body. The estimated 20,500 genes that exist in almost every cell of our bodies is our equivalent of the software we use on our computers every day. And just like our computers, when there is a bug in one of those thousands of genes, it can wreak havoc across the human body. The human body has 23 pairs of chromosomes, one set from the father and one set from the mother. The chromosomes are supercoiled strands that are very tightly packed into the nucleus of a cell. They're so tightly packed that if you were to take one cell and unravel the DNA, it would stretch over 6 feet in length. And this begins the introduction of problem number one. To demonstrate this issue, I wanted to talk about a simple device, a thumb drive. These USB devices released in 2000 were the answer to overcoming the limitations of floppy disks. They were faster, no moving parts, and did not have issues related to surface scratches. The initial devices could store roughly 8 megabytes, or 3 pictures. If you can remember back to that time, how often you would struggle transferring data from machine to machine. You might want to move pictures from your desktop computer to your laptop, and 9 times out of 10 it'll tell you that you've run out of space on your thumb drive. As of 2017, these devices, while used less and less with the introduction of cloud data storage model, have sizes up to 2 terabytes or 2 million megabytes, approximately 800,000 pictures. Gene therapy technologies are on a similar cusp as it pertains to the size of the vessel that a corrected genetic material can be packed into. So a common approach in gene therapy today works like this. Scientists take a naturally occurring virus. They replace the genomic contents of that virus with the corrected human DNA strands that they would like to introduce to a damaged or improperly working human cell. These viruses are referred to as vectors. They are replicated by scientists and can reach numbers in the trillions of vectors within one injectable solution. So a typical approach is to take a vector or virus, then pack the corrected DNA sequences into the vector. Then the vector, just as a virus normally does, latches onto the human cell and delivers the DNA strands by infecting the cell. As stated earlier, the double helix are strands of DNA made up of matching nucleotide base pairs. The human genome is so complex and large that there is no vector that can contain large pieces of DNA beyond roughly 7,000 base pairs. So in other words, only 2 to 3 genes could be theoretically corrected in a treatment. In many illnesses, this is adequate. You might have a single gene improperly working, and these methods provide a clean delivery system. But in a very typical Wolff-Hirschhorn patient, for example, they could have roughly 8 million base pairs deleted. So just as a thumb drive had to increase its capacity to accommodate growing data demands, a vector also needs to be capable of encapsulating millions of base pairs and delivering that payload to all the affected cells in the human body. And this brings us to the second problem. Once the size issues have been resolved through possibly developing new viruses, in combination with compression or zipping of large payloads, the next problem poses big challenges. We've all been affected by viruses on our computers. Most of us have virus software on our computers these days, whether it be a paid subscription or a free service through Microsoft. The goal of these applications are to search and quarantine applications that have been cited as a virus from a list of known bad programs. Of course, what many of us find is that these virus applications don't always work and often stop good applications from running on the machine. So we have to tell the virus software to ignore certain scanning of applications, and the problem is resolved. In delivering gene therapy to a patient, the immune system is similar to a computer virus scanning software. It's very smart and will kill a vector or virus that has invaded the body. The immune system identifies the vector as an intruder by looking at the outer protein shell of the virus and destroys it. So the current treatments for gene therapy have one major hurdle to overcome: the immune system. As a result, the initial treatments delivered to a patient only transfer their payload to roughly 30% of the affected cells, and subsequent treatments can be rendered totally ineffective. Just as the virus software in your computer has the ability to ignore certain programs, methods will need to be developed to prevent the immune system from destroying the vector before it delivers the DNA to the targeted cells. Once we're able to get through the size limitations and the immune system and get good genetic material into the cell, a new problem arises. Very generally, Microsoft Windows has predetermined places for your files. For example, the My Documents directory is designed as a personal area where you can store non-shared files. One of the nice things about putting it there is that it provides an easy access and protects your files from unauthorized access from other individuals. You could store a file in a custom directory, but you don't have these added benefits. A somewhat similar issue arises when placing new genetic material into a cell. Scientists are able to introduce DNA strands into a nucleus of a cell and have them effectively live on their own. So these DNA strands produce proteins and work as intended. But the one big drawback to this approach is that these DNA strands are not being regulated by the chromosome itself. Between the hundreds of genes along the DNA strand of the chromosome, it was thought for many years that this was junk DNA. Scientists could not understand the purpose or function of this junk DNA, or in other words, DNA that doesn't produce proteins. But recent studies have shown that these sequences are actually regulating the expression of the genes along the chromosome. It is instructing the genes to turn on or off depending on conditions. Thus, the chromosome is never over or under producing proteins. As a result, for a patient that has a terminal deletion to a chromosome, it is important to interlace the newly introduced DNA strands onto the terminated chromosome. This way, these new genes will be regulated by the chromosome, thus keeping the homeostasis of the cell intact. The challenge here is to use methods in nature that will force the newly introduced DNA to attach and interlace to the terminated chromosomal end, thus placing the genes where they were intended to be. I wanted to talk about one final concept as it pertains to the entire delivery system, and that is quality control. For a software developer, we're constantly testing and ensuring quality control of our software. Having one mistake will introduce the famous application crashes or warning messages. Because all patients will have different treatments due to their variability of the deletions, it is critical that each vector that is introduced into the body of a patient be checked for accuracy. Scientists are able to determine the base pairs that have been deleted from a chromosomal arm by comparing the good chromosome to the deleted one and taking the delta. One possible approach to ensure good quality control for the trillions of cells in a single treatment is by using dye labeling or fluorescent dyes for identifying nucleotide base pairs. A computer could then scan to ensure that the proper DNA sequencing are created and available in the vector before delivering the treatment to the patient. Delivering an improperly formed vector to a patient could result in introducing a new bug to the gene sequencing and potentially new problems. So at Liv4TheCure, how do we define the cure? Patients that have the unfortunate luck of being afflicted with chromosomal deletion have implied damage to their body structures. Just as if you were to lose a finger in an accident, missing genes in a cell can have lasting damage. Individuals with missing DNA can have missing organs, missing brain structures, or a whole myriad of ailments at birth. Additionally, as a patient ages and genes are turned off permanently, such as bone growth, there is permanent damage and those genes cannot be turned back on. But that's not the full story. Imagine an individual with a poor immune system whose immune cells could be cured. Imagine a person afflicted with mental retardation that could potentially have cognitive gains. Imagine a person who has low muscle tone and strength gain neurological control and muscle size. With gene therapy, all of this is possible, and more importantly, improving the lives of those that have no options. Thank you for taking the time to watch this video. And from myself and all the participants at Liv4TheCure family, we will honor and respect every dollar in pursuit of the cure. Thank you.